Hello and welcome to part 2 of my lecture on introductory microbiology. This is a lecture that's tailored specifically for my 9th grade honors biology students, but if you're learning some introductory material about microbiology and you stumbled upon this video, well, climb on aboard and hopefully you can learn something. Again, this is part 2. I'll be doing a 3-part lecture on this. Very introductory level material on microbiology. And this again would be suitable for junior high, high school biology students. We're going to pick up here. We're in the middle of a discussion on the normal flora or normal microbiota that are found living on us and in us. And we looked at different regions where they're found. Well, in those regions, the regions of our body, say the large intestine or on our skin or on our conjunctiva, there are physical characteristics of those regions of our body that will enhance and be best suited for the bacteria that are residing there. So, for example, E. coli is looking for nutrients that we are producing through the process of digestion, and those nutrients aren't available anywhere else except in the large intestine. And so as a result, that becomes the best suited environment for E. coli to live. So you're not going to find E. coli living on your skin under normal circumstances. That's not where they want to be. They want to be where they can get particular chemical compounds and nutrients that are necessary for their metabolism, necessary for their survival. So there are factors that are going to influence where these things can be found that we produce, physical factors, chemical factors, and so forth. But that would be related to what the first bullet shows there, availability of nutrients, and the second one as well, the chemical and physical factors. But also, kind of a negative for the microbiota or the normal flora would be our defenses, the defenses that we possess in order to protect us against particular bacterial species or the antigens that they possess. I mean, our immune system is going to react in some way to these antigens. And in some cases, there's not going to be an immune response, like, say, for example, for the normal microbiota that are within us. There are antigens on there that are, of course, not self-antigens, but our immune system is not launching an all-out attack against the normal microbiota, and that becomes a great question of immunology and the relationship of our immune system and immunology with normal microbiota. So we will launch an attack against some species, but we don't launch an attack against others. And of course, then the question is, why not? And so it has to do with factors of the microbiota and how they're influencing and affecting our immune system, and how much our immune system is willing to allow to pass by as well. So there are a number of factors, and it's a complex system. The immune system is an incredibly complex system, but what I'm referring to is it's a complex relationship between our immune system and our normal microbiota. Why allow some bacteria to reside on us or in us, but not others? And the immune system is, of course, regulating that through different chemical compounds and communication. So that would be related to the defenses of our host. Our age plays a factor as well. As we age, things change within us. Our cellular structure and the compounds that we produce and the hormones that are flowing through us, that changes as we age. So age plays a factor in it. And our ability to defend ourselves against some bacteria that under normal circumstances and in the right locations, they cause no disease whatsoever, but sometimes they can cause disease in other places. And those are called opportunistic pathogens. And as we age, we give these, what normally never causes any problems, we give these bacteria the opportunity to become pathogenic or to create infections or problems within us, and that goes with the aging process. And gender as well. And so gender affects where these microbiota are going to be found. For example, females are 8 times more likely to develop a urinary tract infection than males. So gender plays a role and a factor in that particular infection. but in others as well, because as I mentioned, hormones are chemical compounds that affect other things within the body, and then those things that are affected can affect the normal microbiota. So it's kind of like a domino effect, and gender can play a factor in that. So we'll look at the normal microbiota of our skin. So if we look at these bacteria or these fungi that are on us, they must be able to withstand secretions from sweat and sebaceous glands, and they have antimicrobial properties in them. Sebaceous glands secrete a substance called sebum, which is pretty acidic, and so that lowers the pH of the skin. We produce antimicrobial peptides as well, which get rid of some potentially pathogenic strains of bacteria. Well, some of these antimicrobial compounds contribute to the low pH of our skin. Sweat also contains antimicrobial chemicals as well, ones that are not necessarily directed specifically to kill microbes, but they create an environment that's, not really liked. It's not a suitable environment for the microbes. So the ones that live on us normally have to be able to withstand these chemical and physical barriers, I guess, to many bacteria. For some bacteria, it's not a barrier at all. They do just fine. We produce this protein called keratin, and it's protein found in the skin. It acts as a resistant barrier as well. And so it's really resistant to bacteria because bacteria do not produce an enzyme that is capable of breaking down or metabolizing keratin. It's called keratinase. Fungi, however, do. And so that's why when you speak about skin infections and skin conditions, fungi are culprits of those. Probably more on the skin than anywhere else in the body because they possess this enzyme called keratinase that enables them to break down the keratin and then use it for their cellular functions. So these factors prevent the microbes that are in direct contact with the skin from becoming permanent residents. And so their numbers keep turning over. Some of them never get the opportunity to become a permanent resident, while others, their numbers never be able to, they don't get to epic proportions. The bacteria can reproduce exponentially, and so we have to keep their population in check. And so we produce these compounds, and then we create environments that are not conducive to their survival, at least to the point where they can't become prolific. Excuse me. So I mentioned this guy in part 1, Staphylococcus epidermidis. It's the most abundant inhabitant of our skin, especially in the upper body. 90% of all the normal flora on our skin is Staph epidermidis, and that's what it looks like. And as I mentioned in part 1, that if you look at the naming of it, which is called nomenclature, staphyle means clustered, coccus means round, so it's clusters of round cells, which you can see in that electron micrograph there on the left. And it resides on our epidermis, and so that's why it's called epidermidis. On the right, that's a gram stain of it. It's a gram-positive bacterium, and it's not looking as, I'm looking at that picture now and I'm thinking it's not quite as purple as they normally turn out. Maybe I should find a better gram stain of Staph epidermidis. It's looking more like it's on the gram negative color range, but it's gram positive for sure, and looks purple under the microscope after you do the gram staining techniques. But I guess some of it looks kind of like a dark purple, I suppose. But you can see also the connectivity between them. They've got pili and the structures on the picture on the left that are connecting some of the cells together, and the quote roots that they form, they're called fimbriae, which enables them to adhere to each other and to the substrate or the surface that they're living on. So we'll look at symbiosis, and symbiosis is a term that's, it's prevalent in biology, but we'll look at it specifically in terms of microbiology here. So the term symbiosis is just simply living together. Two organisms sharing the same space, living together, sharing resources, and it can be good, it can be a neutral, and it can be a kind of a negative relationship. So there are three symbiotic relationships, three types of them: mutualism, commensalism and parasitism. And mutualism is a win-win symbiotic relationship in which all participating species benefit. There's no, you wouldn't use the term host with mutualism because a host is meaning that something is deriving benefit from it. And so with mutualism, everybody wins. Commensalism, you have one of them benefiting and another species is neither helped nor harmed. And parasitism is one benefits and the other is harmed. So one benefits at the expense of the other, and the other, in this case, we call the host. And there's a good video on microbial symbiosis. I think it's a TED Ed video. I love TED Ed videos. They're so good. The animations are good. They're a good length. They're usually around 5 minutes or so. Unlike my videos, which seem to go on and on. Sorry about that, but I miss teaching. I miss teaching so much. So I like talking about this stuff because I don't get to talk about it these days since we're in quarantine and I'm sitting at home. And I get to talk to a computer instead of my charming students. So anyway, sorry, off topic. That's a good TED Ed video on microbial symbiosis. So if you want to, of course you can't click on it, but go to TED Ed and do a search on microbial symbiosis and you'll find a decent video there. So mutualism, as I said, one benefits and as does the other. So they're both benefiting together. This is a mutualistic relationship. Classic example in microbiology is E. coli and us living in our large intestine. So they're synthesizing vitamin K and some B vitamins, and they produce in abundance. And remember, vitamins are nothing more than just chemical compounds. They're complex chemical compounds, but they're just held together by covalent bonds, and they're made up of a few elements, mainly carbon, and so these vitamins then are useful in some way to organisms, whether it's carrying out some kind of chemical reaction, speeding it up. It's being used as an enzyme cofactor, for example. These vitamins are used in a variety of different ways. And what E. coli does is it produces these chemical compounds, and it produces them more than they need. And so they actually they secrete it. And then those vitamins, these chemical compounds, get absorbed into our bloodstream to our benefit. The large intestine, in return, our large intestine, provides nutrients needed by E. coli. So we produce things for them that they need, and so they're benefited. So this is a win-win situation. Everybody's happy, and we can all get along just fine. And you say, but wait a minute, Tinker. E. coli outbreaks, you get it on lettuce or something like that, and people get sick. It's in hamburger. What's going on? You're saying E. coli is a good guy, but yet we hear about these outbreaks. Well, there's different strains of E. coli. And there's a pathogenic strain of E. coli that produces a toxin called the Shiga toxin. And the Shiga toxin will cause pretty severe diarrhea, in some cases dysentery, which is bloody diarrhea. Sorry, I hope you don't have an image of that, but it's very unpleasant. And it's called traveler's diarrhea as well, because people pick it up in different countries and then they travel with it. And it goes with them and they end up having these terrible bouts of diarrhea. The E. coli that is in our large intestine is not the pathogenic Shiga toxin producing strain. And so it's not causing problems there. Now, this E. coli, however, when it leaves its environment that it's in, it can cause UTIs, and I talk about that later, of how that happens. But not here. Commensalism. Commensalism, one organism benefits and the other is unaffected. And this would be like much of our normal microbiota. Of course, E. coli being an exception there. That's mutualism. But commensalism would be, you've got these bacteria living all over our skin. Are they giving us something directly? No. Are we giving them something? Yes. And so they're benefiting from us, but we're not necessarily benefiting, so we don't want to say this is mutualism. However, they are providing us an indirect benefit, and I mentioned this in part 1, where they are colonizing us. And so as a result, pathogenic bacteria cannot colonize us because they're taking up residence there and they're protecting their turf. They're saying, this skin is mine, not yours, go away, and if you try to take up residence, I'll start producing junk that you don't like. And so they actually compete. There's competition going on, as there is in all the biological world among organisms. Competition. And so even this is going on our skin at the micro level. So these would be, like I said, on our skin, and some that are on our eyes as well, on the conjunctiva, that very, very thin layer of tissue that covers the surface of our eyeball. Parasitism, one organism benefits at the expense of the other, and so that the other is the host. In some situations, normal flora can make us sick, and that's when they leave their habitat. And if you skip down to the bottom there, that's an opportunistic infection. So for example, E. coli, they leave their habitat of the large intestine, they gain access to other parts of the body, such as the bladder, the lungs, or the spinal cord. You can develop respiratory infections or meningitis as a result of this, and abscesses and UTIs, urinary tract infections. So they're given the opportunity to cause infection. While they're in the large intestine, everybody's happy, no problem whatsoever. But out of the large intestine, then we can run into some issues. And I thought this was an interesting TED Ed video here, mind-altering parasites. It's not necessarily having to do with opportunistic infections, but it has to do with parasites altering the minds of their hosts. And I use the word mind in quotes. This is parasites altering the quote minds of insects, for example. Where some ants can be taken over by these parasites, or the mosquito is taken over by the malaria parasite called Plasmodium, and so they cause them to behave in ways that they normally don't. So you want to watch a good video, go to TED Ed, type in mind-altering parasites. It's a good one. As nearly all TED Ed videos are good ones, at least the ones on biology. Anyway, I don't venture out to the other subjects. So nutrients, what do they want? Bacteria can colonize only where they can be supplied the right nutrients. And they come from excretory products of the cell, stuff in our bodily fluids. They will consume our dead cells. Like, for example, some bacteria living on our skin are benefiting from all of our sloughed off dead skin cells. They're getting nutrients and energy from that. And food in the gastrointestinal tract, like E. coli, for example, living there getting nutrients from us. What are some chemical and physical factors that can affect their growth? So nutrients can affect their growth, but temperature can. The ambient temperature outside, the temperature of the body, the pH of the environment that they're living in, available oxygen and carbon dioxide that's available to those microbes, salinity or the salt content, and sunlight, specifically the UV rays, which those are all factors that will affect their growth in one way or another, whether benefiting them if it's their optimal temperature, for example, or hindering or retarding their growth if it's not the optimal temperature. Some mechanical factors that do that. And so mechanical meaning movement. Think of things rubbing against each other, or like the chewing action of teeth, for example, is a great mechanical example. So certain areas of the body have different mechanical factors that can affect them from colonizing. And so we chew with our teeth, and then that'll dislodge microbes. And that will prevent them from coming into different regions. We don't swallow them as a result of that. So that chewing action of our teeth can move microbes along, and then we swallow them. They go to the stomach, the acidic environment in the stomach, and the enzymes break them down. So do you want to chew gum? Is that beneficial? Well, yeah. I mean, if it's sugar-free, I mean, you chew gum that has sugar in it, you're just adding sugar to the bacteria that are in your mouth that can lead to tooth decay. But you're chewing sugar-free gum, that chewing action will dislodge microbes, move them off the surface of your teeth. You're going to create more saliva. You're going to produce more saliva as a result of chewing. And so that's going to wash them off the surface of your teeth, and you'll swallow them, and moving your tongue around. And peristalsis, for example, in the intestines. In the small intestine, the microbes never have a chance to colonize there because we're constantly moving food through it. There's continual peristalsis going on in the small intestine. And so it's pushing the microbes through. Once you get to the large intestine, peristalsis slows down big time, and now bacteria have the opportunity to colonize and to grow to enormous numbers. So again, these mechanical factors, they can dislodge the microbes and move them along and send them on their way, and so they never get the opportunity to colonize. So how do bacteria make us sick? So is it them themselves that are making us sick or what they produce? Well, as you can see, it's what they produce. And these substances that they produce are called toxins. And these toxins, and you've heard of the word toxic, toxin, you know it's not a good word. It's not something that, if you describe someone as being toxic, you're not paying them a compliment. If you say, hey, Jim, he's real toxic, you're not saying he's a great guy. No, Jim is, there's something up with him that's not very pleasant. And so toxins are things that are not pleasant. And there are two broad types, exotoxins and endotoxins. And exo, as its name implies, the prefix exo means to exit, to come out from. So exotoxins are proteins that are coming out of or secreted by bacteria. And they're products of their metabolism. And these are some characteristics of exotoxins, generally produced by gram-positive bacteria. Now, there are some gram-negatives that produce exotoxins as well, but by and large, gram-positive are producing the exotoxins. And some of the deadliest substances known to man, the Clostridium botulinum toxin, is the deadliest substance that we know of. Natural or synthetic, it is the deadliest substance. It's a bacterium called Clostridium botulinum. And you say, wow, have I ever encountered this? Well, it's a common soil bacterium. Fortunately, it's an obligate anaerobe, which means once it's exposed to oxygen, it's dead. Oxygen is very lethal to this species of bacteria. Maybe you've heard of Botox injections. That's what it is. We're taking the C. botulinum toxin and we inject it into skin, and we use it cosmetically and also in a therapeutic sense as well, where we try to help alleviate symptoms of this condition called myasthenia gravis, which is uncontrollable relaxation and contraction of the ocular muscles. But anyway, sorry, that's going a bit too much in depth. I don't mean to. But anyway, Botox, BO for botulinum, and then tox for toxin, the C. botulinum toxin. Some examples are food poisoning, and I put poisoning in quotations because technically it should be called a food intoxication because you're taking in the Staphylococcus aureus toxin. And that's the number one culprit for food poisoning, is the Staph aureus toxin. So really you're ingesting a toxin, and so that's an intoxication, but I'm just being picky about this, I guess. But I like technicalities, and I think we should stick with them. So food intoxication, I suppose, is a better term, but we say food poisoning. TSS, toxic shock syndrome, which is also caused by Staph aureus, number one culprit. A few other bacteria can as well, but Staph aureus does for sure. Tetanus, botulism, diphtheria, gas gangrene. These are all diseases and intoxications that are caused by exotoxins. Endotoxins are components of the bacterial cell wall. They're actually in the cell wall, and that's why they're called endotoxins. Generally gram-negative, and there are really no specific diseases with this that we have names for, but it causes fever. Generally, you don't get fever with exotoxins. With endotoxins, you do. Hemorrhaging, so it causes mass vasodilation and leakage of blood out of blood vessels. Shock, which is uncontrollable decrease in blood pressure. It's sudden and it's uncontrollable. And the reason why is because of this severe, what we would call systemic vasodilation, where blood vessels are just opening up throughout the body. And so as a result, the blood pressure plummets. Miscarriages of infants, and it can lead to coma and then death, of course, as well. We call this usually gram-negative sepsis, or sometimes it's just called plain old sepsis, or septic shock is what it's called as well. Again, not a specific disease, but these are all deadly conditions that are caused by endotoxins. And again, generally from gram-negative bacteria. And there are different types of these toxins as well, which I'm not going to get into because, again, this is introductory. Some of the defenses that we have against microbes. Well, we have activated cells that kill microbes. We have macrophages that can do this. Excuse me. These macrophages will ingest these microbes, will break them down using enzymes that are contained within lysosomes inside of these macrophages. We have cells that will inhibit their growth and prevent their numbers from reproducing and getting to epic numbers to prevent them from reproducing exponentially. We have cells that will surround the microbes and prevent them from adhering to cell surfaces. We can neutralize toxins as well. There are, say, for example, these specific groups of antibodies called antitoxins that will surround toxins and prevent them from being absorbed into, say, mucous membranes or the bloodstream. And so they just kind of tumble along, and they never enter the circulatory system or the lymphatic system. And so then the toxins never have the opportunity to do damage to us. So there are a number of different ways that we can take care of these toxins and the microbes. If our innate defenses, meaning the ones that we're born with, if they're not sufficient, then we can take antibiotics to help us out. So pre-antibiotic age, so antibiotics were discovered by Alexander Fleming in 1928, and they weren't mass-produced until the 1940s. But in the age before antibiotics, that's really all we had to rely on were our innate defenses. And we tried all these different ways of trying to fight these diseases, and of course they didn't work. We would do leeches, for example, or bloodletting, and we thought our blood was bad, and so we had to get rid of the bad blood so that it was out of our body, and then we wouldn't be sick anymore. And actually, we were worsening the situation. So anyway, again, off topic, sorry. I do it all the time. My students are all nodding their heads right now. Yes, Tinker does this all the time. Sorry. Antibiotics are taken to assist that. So the antibiotics are using, these are chemical compounds that are, the number one target of which is the cell wall and the plasma membrane of the bacteria to break them down and then to kill the cells. However, many species of bacteria become and are becoming antibiotic resistant, and that's a major problem as well. And that's a whole other topic that I could go on about with the antibiotic resistant issue that we're dealing with currently in the world today. This is not just a United States problem. This is a global problem, and it's at a global scale. And there are solutions to it that we need to explore and research and develop. One of them being phage therapy. But anyway, these antibiotics can be taken in order to assist our innate defenses. These are two videos. Oh boy, I forget if they're TED-Ed or not. I think one of them is Kyrgyzstan. I think that's how you pronounce it. Two good videos on how antibiotic resistance has come about and why, and then why do we feel sick? What's causing us to feel ill? These are two good videos there that unfortunately I can't remember where you can look for them, but I suppose if you went on YouTube and typed in these terms, you could probably find it. So biofilms, last major topic of this particular segment of the lecture. Biofilms are a big deal. They're very important, and they're something that all students should learn about it if they're encountering any instruction in microbiology. You got to learn about biofilms. So biofilms are masses of microbes that are attached to a surface, and they greatly increase pathogenicity. Pathogenicity is just a fancy term for the ability to cause disease. About 70% of all infections are due to these biofilms. So what is it? Well, you can see kind of the picture of it there in that pipe. In the picture on the left, that's a water line, and a biofilm is formed within it. And what all that white goopy stuff is, within that are going to be billions and billions of bacteria, and probably, I don't know, a few other microbes as well, but definitely the vast majority are going to be bacteria. And as a result of living in a biofilm, they will produce this slime, and it appears white. And it's sticky. So this is a very viable thing here that we're looking at. This is not some inert, non-living white goop. It is white goop, yes, but within it are plenty of cells. It's a very vibrant, active group of organisms in what we call a biofilm. So what do? Well, they connect with each other. They communicate with each other. They share nutrients. They share DNA. and they encourage each other basically. Now, biofilms, and I should have mentioned this when I started, it seems like, wow, biofilms are really bad. We don't want biofilms. Get rid of all biofilms. No, biofilms are absolutely necessary out in the environment. And even within us as well, we have very beneficial biofilms, and for sure, most biofilms are beneficial. It's kind of like most bacteria are beneficial. We just pay attention to all the negative stuff. We need to stop being so negative. Let's be more of a glass is half full kind of person, right? Let's all make that decision. We're all a glass is half full person. Okay? Good. Now let's move on. There also is a glass is half empty viewpoint as well. Biofilms are bad when you have pathogenic bacteria. So on their own, the bacteria aren't going to do anything to us. So let's say you get a pathogenic bacterium in you ingest it, or we'll say breathe it in, because that's the number one way they get into us, through our respiratory tract. So you take in a few pathogenic bacteria, you breathe them in, and what's going to happen to you? Well, nothing, because if it's only a few dozen or a few hundred of these bacterial cells that are coming into you, you are an incredibly enormous host compared to these bacteria. So they're going to do nothing. They basically, kind of want to stay hidden under the radar. What happens then is we get an infection and get sick. What happens is the small number of bacteria, they start to reproduce. They start to increase their number. They start to attach to each other, and they start to communicate with each other. And when that happens, now you've got the beginnings of a biofilm. So think of it like they get into you, but they have no idea that there's anybody else out there in the vast expanse of your body, until they start to reproduce and produce more of themselves. And they send out these chemical signals out into the darkness of your body. And these chemical signals may or may not get picked up by other bacteria. If there aren't many bacteria there, these chemical signals won't get picked up, and then they will not be able to communicate with each other. And so they kind of receive the message, hey, I guess I'm the only one here, so I better stay quiet. Whenever their numbers get large, massive, those chemical compounds will get picked up by other bacteria, and then they will be like, oh, hey, I guess I'm not the only one here. And so they'll start to send out pili, and the pili will go out into the dark. Remember, bacteria can't see, and they can't talk. So this is just, they have this, and I'm using these terms kind of loosely. So they, quote, realize that there are other members of their species out there. So they'll send pili out, and they'll start exchanging DNA to support and strengthen each other. They'll start producing compounds that they normally don't produce on their own, and they'll start making this kind of three-dimensional mass of cells. And outside in nature, they do this all the time, like on a stream bed, for example. The rocks are all slippery. Well, why are they slippery? They have biofilms on them. The bacteria are growing on them, and they've produced this mass of cells, and we call that a biofilm. So it really strengthens them. And then, if they're pathogenic, that's when they launch their attack. They start producing their toxins because they, quote, again, realize that there are large numbers of them. And so they will start to produce pathogenic toxins, and then that's what makes us sick. In the terms of the negative biofilms, the glass is half empty biofilms. Okay, these are the bad ones. Plaque, for example, that's notorious one. That's not very good for our teeth. Plaque is a biofilm, and it's a conglomeration of bacteria that are growing together. They're sharing nutrients, and they're using the sugars that we consume and that we eat that are then coating our teeth. They'll convert it into a sticky kind of gummy polysaccharide called dextran, and they will live within that dextran, and they'll use it for their metabolism, and then they start multiplying and dividing and multiplying and dividing. Eventually it gets to the point where it doesn't come dislodged off your teeth. Your toothbrushing, of course, helps, but eventually the plaque becomes so tenacious that toothbrushing doesn't get it off, and so that's why every 6 months you should go to the dentist and have the dentist or the hygienist get that plaque off of your teeth and scrape off the tartar with a tartar pick as well. So these are biofilms that form on our teeth, and it's a conglomeration of a variety of species of bacteria along with other compounds that enhance and enable them to grow into these massive numbers. They have a very complex chemical composition, the biofilm does, because they're producing these compounds that they normally don't produce when they're alone. And so they're making these compounds, and these chemical compounds are interacting with each other and they're bonding with each other. So there's a lot of chemistry that's going on here as well. And so as a result of that, the biofilm develops resistance to our immune systems, disinfectants, antibiotics. I can't even touch it. About 1,000 times more resistant. And so the biofilms then protect the bacteria in this way. Significant problem on internal medical devices like catheters, for example, that are used to evacuate urine from the bladder in people who are bedridden, for example, or even IV catheters. If you're getting an IV that needs to stay in for a long period of time, they'll put a catheter into your vein so they don't have to keep jabbing you again and again. Well, biofilms can form on these catheters, stents that are put into blood vessels to open them up. These stents then become a surface on which biofilms can form. Mechanical heart valves as well become a great surface for them to form on, and contact lenses. They can form biofilms on those as well. So be sure to be cleaning contact lenses and making sure that you are keeping them very clean and using sanitary practices with your contact lenses. So the biofilms don't form. You don't get, for example, pink eye, which is really not that bad, I suppose, but it can develop into something worse if it's let go. And as I said, they communicate with each other. So there are ways that we can prevent them from communicating with each other. We can clean surfaces with disinfectants so that their numbers can't get prolific, for example. Or we can inhibit their ability to talk to each other. If they can't communicate with each other, then they'll have no idea that there's a large number of them, and then they'll just go on thinking, well, I guess it's just me, so I'm not going to cause any problems. I'm going to stay here nice and quiet so that a big white blood cell doesn't come over and engulf me. If we can prevent them from communicating, if we can prevent them from sending these chemical compounds out into the darkness of your body, or from receiving this signal, then they won't multiply and divide. So some examples, I mentioned plaque, pool algae, shower scum. Another one's called floc, F-L-O-C, and it has nothing to do with birds. It's sewage treatment and wastewater treatment. So we encourage these biofilms there to break down our waste and get rid of it. Here's two videos here. Biofilms explain. Now I remember these ones. This one is a TED Ed, and it's a good one. Go to TED Ed and type in biofilms, and it explains them. And it's not all bad and ugly and gloom and doom. It talks about beneficial biofilms. And then this one, bacterial communication. It's an 18-minute video. The first 10 or 12 minutes are really good. It's done by a professor called Dr. Bonnie Bassler, and she is the chairperson of the Department of Molecular Biology at a university you may have heard of called Princeton University, and she discovered in the mid-90s how bacteria have the ability to communicate, and she does a great TED Talk. So this is a TED Talk, so not TED Ed, but TED Talk, and she gives a great explanation of bacterial communication. And just so my students who are watching this know, you'll need to watch this video, and then I'll have you answer questions about it, because it's a really good video, and you're listening to the person who actually discovered bacterial communication, and so it's a great opportunity to learn something. So this is how they grow. Here's how they progress. They start out as a few cells in stage number 1 there, and if you go across to the right, 2, 3, 4, and so on, you can see how they just become more and more prolific. That greenish stuff, I guess, is supposed to be their slime, and it enables them to grow in a large 3-dimensional mass. Then eventually, at stage 5, the biofilm, at least that particular part of it, becomes too massive to support the growth of all these bacterial cells, and so some of them leave. They get released. Go, bacteria, form a new biofilm, and away they go, and they form another beginning stages of a new biofilm. And so this is how it progresses through the stages of biofilms. And how do the bacteria know to leave? Because they're communicating with each other. They communicate when they're in a biofilm. And so when they receive the message, the signal, they leave. So the biofilm is kind of acting like a superorganism. And you can look that up. Google collective intelligence, for example. It happens not just with bacteria, but it's also common in the insect world, especially among ants and bees. Collective intelligence, fascinating topic. And this is kind of how it works here. It works. The amoeba do this even. They have a collective intelligence. Thousands upon thousands of amoebal cells will come together and form this, what we call a slime mold. And they act as one big organism, and then they'll disperse and break apart into each individual organism. So, cool topic to look up. Collective intelligence, especially with ants and bees, it's fascinating. At least for me, I'm a bio teacher, so I guess it's supposed to be. But look at it for a few minutes and learn something. You might find it to be enjoyable. Which I hope you found this discussion to be enjoyable as well. This marks the end of part 2 of the lecture. I will be then continuing on with part 3, the final part of this 3-part lecture on introductory microbiology. Thank you for watching.